From the minute we are born, we start experiencing new things, like opening our eyes for the first time, taking our first steps, reaching for our first toy, and speaking our first words. During the course of our life, we also experience new things, like feeling the sun and air in our hair. Along the way, we might meet new people who like what we like, who like us, thus developing friendships and relationships that will last a long time, like celebrating your birthday along with people you love, who love you, make you feel safe, and are happy to celebrate each milestone accomplished by every year you have life. We love to do things with our body along the way. Some people like to dance, some people like to play tennis, some people like it because of the people they get to do it with. Some people like to play baseball, and others might like swimming. But we get to do these things because of our body, because we get a functioning body that allows us to experience life by doing all of these things. However, for a person living with Parkinson's disease, these simple yet profound moments that define our journey in life, shaping who we become, can get marred by the daily challenges that this condition presents. Imagine the life with the simplest tasks like pouring a cup of coffee, buttoning a shirt, become monumental feasts. Parkinson's disease, a progressive disorder, affects millions of people worldwide, robbing them of their life experiences and of their ability to live life all the time to the fullest. Parkinson's disease is a neurodegenerative disease that affects millions of people worldwide. It is characterized by the progressive loss of dopamine producing neurons in the brain. Dopamine is a neurotransmitter that is essential for motor control, so as dopamine levels decrease, people with Parkinson's disease experience symptoms such as tremors, rigidity, slowness of breath, and balance problems. In Guatemala, the Parkinson's disease is approximately 100 cases for 100,000 people. That means that every 1,000 people in Guatemala, at least one has Parkinson's disease. And according to the study by the National Institute of Statistics in Guatemala, the prevalence of Parkinson's disease in the country has increased from 71 cases per 100,000 to 100 cases per 100,000 in the last decade, and the average age for developing Parkinson's is 60 years old. Currently, there is no cure for Parkinson's disease, but there are treatments available to help manage symptoms. However, these treatments can cause side effects that do not stop the progression of the disease. This is where gene therapy comes in. Gene therapy is a promising new approach to treating Parkinson's disease. It involves delivering genes to cells to correct genetic defects or to introduce new genes that can produce dopamine. Our project is focused on using gene therapy to produce dopamine in human neuronal progenitor cells derived from Parkinson's disease patients. To do this, we will use a lentiviral vector to deliver genes encoding the enzymes involved within the dopamine biosynthesis pathway. These enzymes are tyrosine hydroxylase, GTP cyclohydroxylase, and aromatic amino acid decarboxylase. If our project is successful, it could lead to the development of a new gene therapy treatment for Parkinson's disease. This treatment could help to improve the quality of life of people suffering with this disease by reducing their symptoms and slowing the progression of the disease. We are Ictancanik, and we urge you to join us on this journey as we explore the incredible advancements and breakthroughs that promise to change the life of those living with Parkinson's disease. This is our project.
For our design, we decided to use the plasmid PRRL-PPTSFFVHA-RUNX1CIRES-EGFP as the vector for our design. The parts added to the vector were the sequences that code for the tyrosine hydrolase that will be referred to as TH, also FTP cyclohydrolase 1 that will be referred to as GTP-CH1, and the aromatic amino acid decarboxyhydrolase that will be referred to as AADC, which are the enzymes involved in the biosynthetic production of dopamine. The sequences were introduced after the RSV promoter that is widely used to provide high levels of expression in different cell types. To optimize our design, we decided to add a P2A sequence between each sequence of the TH, GTP-TH1, and AADC enzymes. These two A peptides induce ribosomal skipping, so that when these peptides are inserted between two CDS, the ribosome translates the sequence as two proteins that will fold independently. To build our plasmid, we propose the use of the Git assembly method, since there are several parts that have to be assembled and the amount and size of the parts allowed us to assemble them in a single reaction. We want to introduce the part in the MUN1 region of the MCS of the plasmid, since the enzyme will cut the plasmid only in that position and does not cut our parts and other sides of the vector. After having our vector ready, we propose to introduce it in a 293T cell line that is commonly used for viral production, and its genome enables the cells to produce recombinant proteins within plasmid vectors containing RSV promoters. The plasmid includes sequences for ampicillin resistance, which will be helpful to select the transformed cells, along with the blue-white screening method. It also includes a GFP sequence that will be also helpful to select transformed cells and in the vector titering. Also, a PSPAX2 and a VSVG plasmid will be introduced. The first one includes GAG, POL, and REV sequences, important for the packaging of the virus, and the second one contains the sequences used for the envelope glycoproteins to form the virus. To test the results, we propose to verify the plasmid expression in the ACS5001 cell line, which are neural progenitor cells used for the development of treatment for Parkinson's disease. We will measure the infection of the virus and the plasmid expression by PCR to confirm the introduction of TH, GTP, CH1, and AADC sequences. Western blot to confirm the production of the enzymes, and HPLC to quantify the dopamine concentrations that the cells produce. After modifying and creating the PRRL PPT as FFVHARUN X1C IRES plasmid, we obtain a valuable set of bench-linked metadata that translate into the sequence map. This map provides a detailed view of the genetic information of the plasmid, which is essential for understanding its operation. Initially, we export a FASTA file representing the complete nucleic acid sequence of this modified system. To analyze and better the understanding of the functioning of the modified plasmid, we employ SoftBerry, a powerful bioinformatics software tool. SoftBerry is versatile and specializes in gene identification, functional signal analysis, and gene function determination and interpretation of gene expression data, among other functions. This software provides customized solutions for genome analysis and comparison, making it particularly suitable for predicting the operation of the modified plasmid. Within SoftBerry, we use the gene prediction algorithm FGENESV, which is an automatic annotation package. FGENESV not only predicts genes but also identifies promoters, terminators, and operants, in addition to automatically annotating the predicted genes by comparing them to databases, such as COG and NR. This software package is a powerful tool for understanding the structure and function of transcriptional units in our plasmid. As a result of implementing the FGENESV algorithm, we generate a multi-FASTA file containing all the information related to the prediction of our functioning plasmid. This file reveals that the total sequence length is of 11,964 base pairs, and a total of 9 genes have been predicted, equivalent to 9 identified transcription units in the plasmid. This information is essential for a deep understanding of the behavior of our modified plasmid. BLAST is a program for aligning sequences, whether DNA, RNA, or proteins typically used for local sequence alignment. It compares a given sequence to multiple sequences found in a database. One of its extensions is BLAST-TP, which compares protein sequences against the NCBI database, providing alignments and information on the closest matching sequences. All predicted genes demonstrate their synthesis basis on the functionality of the entire plasmid. In this case, working with the PRRL, PPT, SFFB, HA, RUN, X1C, IRES, EGFP plasmid, and performing a blast search against the resulting protein sequences yields the gene list presented in the table of synthesized genes in the process. All of them exhibit an accuracy greater than 75%, so the variation can be minimal. After conducting a thorough analysis of the plasmid essential to the project, it is evident that it can predict the production of the three key syntheses. The highest scored genes include tyrosine 3 monooxygenase, which is responsible for the dopamine biosynthesis, GTP cyclohydrolase 1, isoform 3, which operates in the metabolic pathway for BH4 synthesis, a cofactor that plays a crucial role in the biosynthesis of various neurotransmitters, and EGFP, which is practically identified as a synthetic construct. Consequently, it's apparent that the created plasmid proves to be an essential candidate for this design system.
But to bring our project to the public and help society, it needs to go through a bioprocess engineering process, and we will tell you how we will do it next.
After experimental data has been collected of the modified ACS5001 cells growth, a kinetic growth model can be developed. There are different types of kinetic growth models used to describe a specific cell line's growth rate, where one of the earliest and most used is the monad model. Kinetic growth models are useful because they assist us engineers to design and control biological processes. We propose to use the monad model to obtain kinetic parameters such as μmax and Ks. Here is the monad model, where mu is a specific cell growth rate, x is the cell concentration, t is time, mu max is the maximum specific growth rate, s is the substrate concentration, and ks is the substrate affinity constant. To determine the volume of the reactor, we propose using the following equations to calculate space-time, represented by the variable tau. Multiplying tau by the flow rate, we obtain the volume of the reactor. The equation is for a batch reactor, also known as STR, and it must be evaluated if this type of reactor is the best for the process or another one might be better. The equations used to obtain space-time for a batch reactor are the following integrals, where Ca is the final concentration of substrate and CaO is the initial concentration of the substrate. In order to solve these integrals, you can use the experimental data collected and an online simulator such as Berkeley Madonna. In order to solve these integrals, you can use experimental data collected and an online simulator such as Berkeley Madonna that will allow us to find the space-time for a batch reactor. When we have this information, we can calculate the volume needed for scaling our process into a bioreactor.
For us as a team, it's clearly important to collaborate with our community, so our human practices efforts have been fundamental in spreading the word about this groundbreaking research. First and foremost, we've had the privilege of hosting hands-on workshops for high school students. These workshops serve as a getaway for the exciting realm of synthetic biology, allowing students to roll up their sleeves and dive into DopaSyn project themselves. But we didn't stop there. We actively sought feedback from both students and their parents, ensuring their project resonates with the community. Collaboration and responsibility are key in our journey. We engage in meaningful discussions with the Ministry of Public Health and Social Assistance and the Universidad del Valle de Guatemala's Animal Ethics, Youth and Care Committee. These organizations help us lay the ethical and safe foundations for our research. Biosafety Level 2 protocols, containment precautions, and the meticulous management of biohazardous waste and rigorous personnel training were in use during this project because safety and ethics are our guiding stars. Last but not least, our interactions with the National Biotechnology Commission have been invaluable. They've offered insights into the potential applications for our research, reminding us the broader impact of the DopaSyn project. They also invited us to participate in the annual National Biotechnology Congress, and they also provided us with feedback for our project. They said that animal testing should not be included in our project because it would require the project to be stopped for review by the Ministry of Public Health and Social Assistance and all entities associated with the university. We decided to follow this advice and we looked for the option of simulators. We now propose that in the future, if tests are to be carried out, they should be in a simulated cellular line. We also get feedback from the community to ensure that our project was still within the community's best interests and aligned with its needs. For example, they asked us about the microorganisms or bacteria that we would be using for genetic modification, and if these organisms had any success of curing Parkinson's disease. As far as the scope for our project goes, they suggested that we focus only on one condition at a time. So rather than treating Alzheimer's and Parkinson at the same time, we decided to focus only on Parkinson's disease, taking this feedback into account. We formed a particularly close partnership with the UNAP team from Chile. We share our genetic circuit with them, and they gave us valuable suggestions on how to improve it. We also guided them in their human practices, which are important for ensuring that synthetic biology is developed and used in a responsible and ethical way. This year, we wanted to do something different with the artistic section. We wanted to create something that was very original and creative. We decided to hold a contest between the team members and the students from different departments of the university. The contest consisted of describing our project to an artificial intelligence after a lecture and then sending the image to the emails team. We received over 35 submissions and were blown away by the creativity and the talent of participants. The design team selected the image that best captured the essence of our project, and we used that image for our project website. Thank you for listening to our project. We are hopeful that it's going to help improve the daily life of those suffering with Parkinson's disease.